I think that one of the beauty of, even though I'm a proteomics person, and so I don't really do much kind of transcriptomics data, but that's the beauty of the field and how it's and the importance of the kind of single cell transcriptomics. And hopefully soon, as the field progresses and the technology progresses, we'll be getting more of the kind of single cell proteomics data as well. But it's really highlighted how heterogeneous these populations are of microglia and Alzheimer's disease. We see massive differences in different populations of microglia. And so we can't think of it as a binary good and bad anymore. That's how the field obviously used to be. But now we really appreciate there are particular subsets of microglia that drive pathology or that drive repair or that have all the other functions. Even in a kind of healthy brain, there are these different populations of microglia. And so it's really kind of appreciating the heterogeneous landscape of microglia and how these populations change during disease. Unfortunately, this means that this now becomes a lot more complicated when we're thinking about therapeutic targets, because previously we would think, okay, all microglia are good at one point, then they suddenly change and all microglia become bad at another point. So if we just get rid of all the bad microglia, we just wipe them away, then we might have, this kind of therapeutic target. When really we know now that there are so many kind of subtle changes. There are populations of microglia that are present in Alzheimer's disease, but we still don't really know if they're good or bad. They're present, but are they benefiting the brain? Are they damaging the brain? Does it change as disease progresses? And so it's really kind of understanding these subtleties and these kind of nuances with the microglia populations. And I think the beauty of that and how the field is kind of progressing in that is we see these at the transcript level and we can kind of get an idea of maybe their function, but that's the importance of getting this data and to the single cell proteomics level, is to understand, are these populations the same? Can they be defined in the same way at the protein level? But with that kind of single cell proteomics data, it will really give us an insight into their functions and to really understand, okay, these populations are present in healthy versus Alzheimer's, but actually what are they doing as well? So that will be the kind of that, and that's how the field is progressing now. We're really trying to develop tools and techniques to define single cell microglia at the protein level and really understand how this kind of complements the transcriptomic data, but also what kind of additional insights this provides to us and gives us more of an idea of the role of microglia in Alzheimer's disease.